Hello cute little children, I hope that you all are doing wonderful, and today I have come up with a one short video of the most important topics for your midterm examination, as we know that your midterm is ongoing, right? So we thought we should be helping you out, as you might be confused between a lot of things, what chapters to study, what to study in a chapter. A lot of questions are there, and this will be exclusively for biology, so all the biology people come together. Let's see what is important and what is not that important. Okay, so the chapters that I'm going to cover today is these many chapters. Now if you think that ma'am, one chapter is missing, two chapters missing, yes, I understand, different schools have different syllabus, so you can tell me in the comment section. I will write down what is important for you. Okay, will that work? I hope it works. So we will go in order of high weightage chapters. So I started with molecular basis of inheritance because it has 13 marks. So your whole paper is out of 70 marks. So out of 70, 13 marks is coming from this chapter. So it's a very important chapter. You cannot leave it by any chance. And let me tell you what is the important topics, and in topics also what is much more important for you to know and how to write and everything. So experiment in search of genetic material. So there were 3 experiments being done in search of genetic material. They were trying to find what is a genetic material. One was Griffith's experiment. So you should be knowing about Griffith's experiment. Okay. Then the other one is Avery and MacLeod experiment. And then you have the renowned, very famous Hershey and Chase. Now out of these 3, I want you to put a star on both of them. Remember the name of the organisms also involved over here. So if bacteriophage is involved over here, what is involved over there? Pneumococcus and mice is involved over there. So all these small details are very much important in the case of experiment in search of genetic material. Okay. Now going ahead, what do we have? We have DNA replication. Now in DNA replication, you should be knowing whether DNA replication is semi-conservative or not. Yes, it is semi-conservative. You get one strand from both the parents, right? One new strand will be formed and one will be parental strand which you got from your parents. So this will be semi-conservative. So you should be knowing about whether it's semi-conservative or not. And if you see through it, okay, you have 2 strands in them. One is leading strand, the other one is lagging strand. Okay, so you should be knowing which one is leading strand, which one is lagging strand. Okay, what do you mean by Okazaki fragments? So there are Okazaki fragments over there. Now there is an important enzyme which is working for DNA replication, that is DNA polymerase. So you should be knowing what is the direction of DNA polymerase. It is 5' to 3' direction. It makes a new strand in this direction. So which strand will be known as leading strand? What will be the direction of this particular strand? What will be the direction of this particular strand? All of this thing, very cute little things are very much important for your examination. Cool, works. Going ahead, we have next thing, that is transcription. Again, here another enzyme is being used. What do you call this enzyme as? RNA polymerase, my dear children. You call this as RNA polymerase. Again, the direction of DNA polymerase and RNA polymerase is the same. It's 5' to 3' direction. Okay. Now you have to study about, there it is leading and lagging strand. Here it is what, children? Template and coding strand. Template and coding strand. So you should be knowing the direction of template strand, you should be knowing the direction of coding strand. Okay, and it says that initiation factor is there. Okay, termination factor is there. Where do they go and attach? Okay, you have initiation factor, you have termination factor. Where do they go and attach? So this location is asked, but always remember the location of these things has high resemblance to the coding strand. So you have to make sure that these things are linked, my dear children. So you should be knowing the location, okay, and it is always in reference to the coding strand. Okay, now going ahead, we have the next topic. Where is the next topic? Why is it not coming? Just a second. Yes, translation and genetic code. So again, the enzymes that are involved in translation, well, there is no such enzyme that you have to study, but you have to know that what happens in the first phase of translation. In the first phase of translation, amino acylation happens. Okay, amino acylation, what do you mean by that? That is also a question that has come. Amino acylation is basically addition of amino acid, okay, and this is also known as charging of tRNA. So amino acylation, when does that come, and then what is the first process that happens in translation? So what is the first phase? That is amino acylation. What is the first process that happens in translation? Basically the first step of translation is when the smaller subunit of ribosome comes on the mRNA, and then the whole thing happens. So that is the first step. So you should be knowing the small details. Now talking about genetic code, you should be knowing about the features of the genetic code. There are certain features given in your NCERT. It is degenerate, it is universal, this and that, okay. So you should be knowing about the features. You should be knowing about the features. You should be knowing which are the 3 stop codons. 3 stop codons, okay. 1 start codon, the name of it. And remember that where is the codon found, where is the anticodon found. So if this is the mRNA, this is where the codon is found. The anticodon is found on the tRNA over here. This is the anticodon. Okay, so remember that as well. So this is from translation and genetic code. Going ahead, we have one more thing, lac operon. Fully, it is very much important. They can ask you to draw the structure of lac operon and also explain it in detail. All the enzymes you should be knowing what they do and explain the process. Explain the process. Why is it known as negative regulation? They'll ask you why is it known as negative regulation. So all these small things you need to be knowing. So these are the things that is from molecular basis of inheritance, which is very much important. I know that chapter fully is being covered, but at least you know what is the most important thing which you have to put your attention in. Sometimes we just study, we keep on studying, we do not know what is important in this particular topic. So that is the sole motive of this particular video as such, okay. So molecular basis of inheritance is done. Now we move on to the next one, that is principles of inheritance and variation. Again, this is of 13 marks. Now, first thing, ratio of monohybrid and dihybrid cross. So you should be knowing that what plant did Mendel chose. Mendel chose pea plant. So you should be knowing about that. What was the F1 ratio of this? What was the F2 ratio of this? And this cross, where only one character is being used, you call this as monohybrid cross. So you should be knowing the ratio of monohybrid cross. What ratio, children? Phenotypic ratio of F2 generation. Okay, genotypic ratio of F2 generation. So phenotypically 3 are tall, 1 is dwarf, okay, and genotypically there is this T and then, so over here genotype as in the genetic makeup. So this looks like one, like same thing, okay. This is, there are 2 of them which has the same genetic makeup. This is, there is only 1. So 1:2:1 is the genotypic ratio of the F2 generation of monohybrid cross. Similarly, you will be having phenotypic ratio. Similarly, you will be having phenotypic ratio of the dihybrid cross also. Phenotypic ratio of dihybrid cross, over here it is 9:3:1. Okay, that is a phenotypic ratio. If you are asked about the genotypic ratio of dihybrid cross, I'm not writing the full thing, but genotypic ratio will be, you can remember, 1:2:1 you write, and then 1:2:1 you write, and then in middle this is 2, okay, and in middle you write double of this, 2:4:2. This will be, works. So this is ratio of monohybrid and dihybrid cross. Please know that. Okay, know how to form it. Okay, make the Punnett score and everything and try to show it. Going ahead, we have exceptions of the laws of genetics. So we have the exception of incomplete dominance. Incomplete dominance. So they will ask you, Snapdragon shows what? Basically, Snapdragon is the example of this. So you should be knowing the example of all type of exceptions. Okay, one is this. The other is codominance, which is shown by the ABO blood group. You have multiple allelism. That is also shown by ABO blood group. All this thing. Okay, so this is very much important. These are the exceptions of the laws of genetics. And again, you have skin color, that is phylogeny, pleiotropy. Also as an example, you can go through that also. Okay. Now, if you have any confusion, you can go watch the videos. If you are worried that, ma'am, I'm not able to understand, put the video in 2x speed and understand. It's very easy. Trust me for that. Okay. Going ahead, you have color blindness. Color blindness is a very important genetic disorder. Okay. Why color blindness is important? Because I saw so many numericals on it. Okay, numericals as in not hard and fast numericals. Okay, they will ask you that mother is color blind, okay, father is color blind, what percentage of child will be color blind? So if mother is color blind, okay, if mother is color blind, then it is XC, both the genes are affected, okay, and if father is color blind, then it is XCY. So over here, whatever you do, all the children will be affected. So it is 100% of them will be color blind. So you should be knowing when does color blindness come and all. Okay, so you have to do a bit research, you have to study a bit, but I am assuming that you know that and you just have to revise it. So color blindness is something that is very much important. Then comes sickle cell anemia. It is also important. Sickle cell anemia and hemophilia. So they have asked once that hemophilia and sickle cell anemia both are genetic disorders. So both are blood related disorders. How they are different from each other? So you should be knowing that this is autosomal recessive disorder. This is X-linked recessive disorder. So how they are genetically different and all, you should be knowing it. Okay. Again, I am not telling you all this to make you panic. No, it's for you to understand that okay, we still have time, we can study it. At least some things, at least if you are not prepared with a lot of things, at least study a few things and go there. Okay, and do not be worried, this is just an exam, you still have your boards coming. Okay, this is midterm examination, you still have your boards coming, and I'm telling you, you're going to rock in your boards. So please don't worry about that, yeah? I feel so bad when I see you guys tensed. I understand you, I was also in the same position once. So yes. Apart from that, you have trisomy and monosomy condition, as in they'll ask you when does Down syndrome occur. So Down syndrome happens when you have one extra 21st chromosome. So this is an example of 21st trisomy, okay. Then they'll ask you what happens in Klinefelter's syndrome. This is XXY condition. There is one extra X chromosome, okay. So that means the number of chromosome altogether will be different. So usually we all have 46 number of chromosome. 46 number of chromosomes are there, okay. Over here we have normal 45 autosomes plus XXY. So the autosomes are normal, the sex chromosome is one that is extra. So it becomes in overall 47, okay, 46, 47, 48, sorry. Sorry Shaktiman, normal cells over here are 44. Okay, 44, and then you have an extra which makes it 47. Now Klinefelter is done. Turner syndrome, what is happening in that? You should be knowing that. So there is X and there is no other chromosome. This is not. There is nothing in there. Okay, this is X naught condition. This happens in the case of Turner syndrome. So this is an example of monosomy. Okay, so you should be knowing about that. Going ahead, what do we have? The 4 contrasting traits. So for this thing, for understanding the ratio of monohybrid cross, you know the P plant was selected. So you should be knowing the names of all the dominant and the recessive traits which were there. Okay, one is tall, one is height, feature is height, and then you have tall and dwarf. Okay, then for example, seed shape, round and wrinkled, seed color, green and yellow. All this thing, all the 7 pairs of contrasting traits is very much important. Okay, I have given you a trick to remember the dominant traits. So this is the dominant trait over here that you need to remember. Green, round, axial as in the flower axial. Okay, V was for violet flowers. Okay, I was for inflated pods. Okay, inflated and constricted, you have one is, that means in your NCERT it is written full. So full means inflated. Okay, and T is for tall and Y is for again yellow color. So these are the dominant traits. You have to know which one, seed color is what, flower color is what. But yes, if it is of any help, you can utilize this. So you should be knowing about that also. Works? Okay, it's easy, my children, you will be able to do it. Now, yes, also just understand or remember this fact that when we say linkage is more, that means if the genes are closely located, they will show less recombination. If the genes are closely located, they will show less recombination. Recombination means this part of the chromosome is going to other part. So when this part of the chromosome is going to other parts, the genes is also going. So they are not giving different characters. The same character, because the genes, both of them, they are going together itself. So linkage, if it's more, if the genes are closely linked, or you say tightly linked, we say recombination is less. Just know that funda in your head. Works? Okay. Next thing. Next chapter we have human health and diseases. It is of 11 marks, starting with cancer. Cancer is very much important. You should be knowing everything about it. Starting with detection. Okay, that is most commonly asked. How cancer cell and normal cell are different. Okay, then next thing that you have is detection. Cancer cell, how it is caused. Causative things. Okay, in causative things, the most important one comes to be proto-oncogenes. So they always ask what are those genes which are already there in human beings but which causes cancer later on. This is proto-oncogenes. Okay, so and the treatment methods. And the treatment methods. That is about cancer. Cancer, everything you have to study. HIV, AIDS. HIV, you should know what cells does it affect. So you know it is macrophage and it is T helper cells. Please children remember it, T helper cells. Majorly it is affecting this cell, it is killing all the TH cells, okay. And then you should be knowing what did WHO do about it. World Health Organization, what did it do? And what is the test for HIV AIDS? For HIV AIDS we have ELISA. So they can ask you to expand this. ELISA stands for enzyme linked immunosorbent assay. It has come in the previous year, which is why I am telling you, okay. Now going ahead, we have malaria, life cycle of Plasmodium, very much important. So you should be knowing what stage is spent in the mosquito's body. Mosquito is basically a vector. Mosquito is basically a vector and a host also. Okay, it is a vector, it carries thing and it is a host also, because over here also Plasmodium spends a bit of its life cycle. And then you should know what stage it spends in human's body. Okay, and why do we have chills and all of this? Because hemozoin is released from the red blood cells. Hemozoin basically means hemoglobin has been feeded by the Plasmodium. When it feeds on it, it breaks heme and globin. Now heme is toxic for it, so it converts heme into hemozoin. Now when the RBC bursts, this hemozoin comes out. This toxic, it try to make some things less toxic, but this is something foreign for us anyways. In our body we have heme. Someone is getting inside our body and converting into something else, and when the RBC will blast off, when these things comes off, obviously we will be having fever and chills and everything. Okay, so that is there. You should be knowing the life cycle properly, and I have made a very good video for life cycle of Plasmodium. If you want, you can see it. It's a very nice video, a separate video for Plasmodium itself because of its high weightage and NEAT also. Okay, now overview of diseases and name of pathogens. Over here you should be knowing the very specific features. For example, in the case of typhoid, they always say 39 degrees Celsius, 40 degrees Celsius fever, high fever is there, sustained high fever. So that is a very specific feature of typhoid. So they can give you a question that this is the condition, tell the disease. So you should be knowing it's typhoid. Okay, so the very common features of all these things. Typhoid. Pneumonia, you have blue nails. Okay, your lips turn blue. Why? Because there is less amount of oxygen in there. So these features are very specific to them. Okay, now if you have rashes, round shaped rashes, that means you are affected with the ringworm disease. Okay, you have constant intestinal bleeding very much. Okay, that means you are affected with Escherichiasis. Okay, now one more thing, the parasites that causes, you should be knowing that Aedes mosquito is causing two different diseases, dengue and chikungunya. Okay, then we have this mosquito, that is Anopheles. What does it cause? It causes malaria. Okay, and then you have Culex mosquito, which causes elephantiasis. So all this thing is important. Different mosquito have different things. All of them, they are female children. Mosquitoes that feed on the blood are females because they want to feed their babies. Okay, it's not babies, basically for reproduction process to happen properly they need blood. This is a meal for them. Okay, so that is very much important. Works. Now going ahead, we have innate and acquired immunity. In that, okay, in all type of immunity, first thing, antibody diagram you should be knowing, the structure of antibody. You should be knowing vaccination. Okay, vaccination. You should be knowing why anti-venom is given to person who have got a snake bite. It shows passive immunity. Okay, it's a passive immunization method. They'll ask you what do you call this method? You call this method as passive immunization. You are not allowing for your body, you are not waiting for your body to make antibodies, you are getting the antibodies from outside. This is known as passive immunization. Okay, so think about that. And allergy, what are the cells that are responsible for allergic responses? Mast cells. that is much important. Mast cells are important, and mast cells get activated because of IgE. It's IgE, okay, that is there. And then lymphoid organs, primary and secondary lymphoid organs, just go through it once. Just go through the primary and secondary lymphoid organs. Primary lymphoid organs are where the lymphocytes, the WBC, RBC, everything is made, and secondary lymphocyte organs are where these guys meet with the pathogens and everything. Okay, so spleen is an example, lymph node is an example of secondary lymphocyte, secondary lymphoid organ, and all those things. Okay, works. So this is important for human health and diseases. Again, I specifically told you what you have to anyway study. I know it's huge, but there is in biology. Can you sum it up and tell that no, this is not important, this is very much important? No, you cannot. I can tell you the most the expected type of questions that have come previously. So that is what I am trying to do over here. Okay. Now next thing, we have sexual reproduction in flowering plants. This is of 11 marks, my dear children. Okay, first is apomixis. They will ask you, apomixis, does it have anything to do with asexual reproduction? Because all the organisms that are formed are like clones, right? So yes, it is like asexual reproduction, but it is not exactly asexual reproduction. Okay, now they will ask you, how is apomixis happen? How does it happen? So please have a detailed study about apomixis. It's a very important thing. So many questions have come. Outbreeding devices, as in the plant itself have restricted itself from doing self-pollination. So there are certain things that it does to avoid self-pollination. You need to know that 5 or 4 points. Okay, it's very much important. Again, questions have come from this particular topic. Pollination, autogamy, geitonogamy, xenogamy. You should be knowing what plants are dioecious, as in what plants have sexes on different plants. So there will be a female plant and there will be a male plant. So over here, which if this is the condition that is dioecious, the plants are dioecious, will they show geitonogamy, will they show xenogamy, will they show autogamy? So autogamy anyway is not possible. Autogamy happens when in the same flower, from the pollen to the stigma it is happening. This is known as autogamy. Geitonogamy means in the same plant you have different flowers, from here to here things are getting traveled. This is known as geitonogamy. Xenogamy means you have two different plants, from here to here things are happening, then that is known as xenogamy, when the plant itself is different. Okay, now if this is a condition where you have one sex in one plant, another sex in another plant, do you think autogamy and geitonogamy can take place? No. Autogamy means this is not happening over here. The other sexes is it's over here, right? So it cannot happen. Geitonogamy, do you think that can happen? No. Again, it's on the same plant that geitonogamy has to happen, from this flower to this flower, but both of them are the same sexes, so again cannot happen. Only xenogamy can happen. So you should be knowing what is dioecious plant, the examples of dioecious plant. You have papaya, you have date palm and everything as an example. PP, papaya, date palm and everything. Okay, now you have monoecious plants. So you should be knowing about their name, maize and everything. You should be knowing about that. Okay, the names of it. It's given in your NCERT. Now pollination, yes, they will ask you that. Okay, they have asked like a flower. They have given a picture of a flower being pollinated by a bee, and they will ask you what type of pollination is this. So you say it's basically insect-borne pollination, right? Now what are the features of the flower which makes it attractive? So you should be knowing the features of the flower, okay, that it has bright color leaves and things like that. So for each character you have different set of features. Please know, for insect-borne, for a water thingy, for everything else you have something to remember, so you have to go through that very clearly. Okay, I hope that is done. Yeah. Now going ahead, we have microsporangium and megasporangium. You would be asked to draw the structures. You might be asked to draw the structures, so please have a clear-cut idea about anthers, sectional view, okay, about the megasporangium, the embryo sac and everything. It's very much important. Know the labelings and learn it. So obviously this topic you cannot miss, okay. Obviously you cannot miss this topic, even it's huge, but it will be good for you. Now double fertilization, they will ask you how is a zygote formed, okay, and how is the endospermic nucleus, the nucleus, primary endosperm nucleus and everything being formed. This is known as double fusion. Why does it happen? How does it happen? Why do you think endosperm comes? Why do you think endosperm is formed before? Endosperm is formed before what, children? It will be formed before the zygote formation too, where the zygote converts into embryo. Before that the endosperm is formed, so that it could provide nourishment. so that it could provide nourishment. That is the answer. So you should be knowing all these small things. Okay, apomixis is very much important. Please do not forget to learn it. Okay, even though it's a tricky thing, again I have made a video on this thing. If you want, you can go ahead and watch. Okay, going ahead with the next chapter, that is human reproduction. Again, it's of 10 marks. Okay, what all do you have to study? Gestation and parturition. In gestation, you have to remember the placental hormones. The placental hormones. So there is HCG, very important hormone, and there is human placental lactogen, everything. So you should be knowing about all these hormones, and there is two more. So there are 4 hormones from placenta. This and estrogen and progesterone. So estrogen and progesterone, always it is there in our body, but still it is released by placenta also. In parturition, you know again the hormones that are released, oxytocin. What happens over here in parturition, what is fetal ejection reflex and stuff like that, you should be knowing. Going ahead, we have fertilization. The site of fertilization is important. Where does fertilization occur, children? Ampulla. Okay, or what changes happens in fertilization, what all processes are involved, and how does the ovum block the entry of many sperms. So this is known as polyspermy. How polyspermy is prevented? This is prevented. That is also a question that could come. So you should be knowing about that. Okay, going ahead, we have menstrual cycle, very much important. Very much important. You have to know the role of all hormones. Role of all the hormones. The 4 major hormones. When does ovulation take place? Basically during LH surge. So you should be knowing about LH surge. Then you should be knowing what happens to the Graafian follicle. It converts into corpus luteum. What does the corpus luteum release? It releases progesterone. What is the role of progesterone? It maintains the endometrium lining. So all this thing is important. Now, if during pregnancy, who takes up the role of LH? It is HCG that takes up the role of LH. HCG will now tell the corpus luteum to keep on releasing progesterone. Usually LH does that, but now over here it is doing. Okay. Now structure of male and female reproductive system. Again, you could be asked to draw it. Don't think you won't be asked. You could be asked to draw it. So you have to know it, the structure very much carefully. Okay. And while you learn this structure, obviously you will be learning about the accessory ducts and everything, which is very much important. The glands, accessory ducts. Okay. Now stages of gametogenesis, especially it comes to oogenesis. When it comes to oogenesis, you will be asked that when is the secondary oocyte formed? In which stage is it arrested? Okay, does the secondary oocyte form the ovum before fertilization, or does it form the ovum after fertilization? Well, it is after fertilization. Okay, or during, basically it's not after fertilization, it's basically when fertilization is happening. At this point of time, the ovum is being formed, the whole completion of cycle is done, and the ovum and the sperm's nucleus will be fusing together. That is the thing. So it's during fertilization that this is formed, so secondary oocyte and everything. So in which stage it is released? Okay, in which stage it is released? It is released in the form of secondary oocyte. Then only during fertilization it will be formed into ovum. So that is very much important. Got it? So that is important thing. And then the different stages of spermatogenesis, they will give you as arrow questions from here to here, here to here, A to B, which order is correct. So you should be knowing the order of this. Works? So this is for human reproduction, what is important. Now going ahead, we have reproductive health, which is the last chapter I have to cover today. This is of 6 marks, my dear children. 6 marks is also important, right? So over here we have few topics which are very much important. The chapter itself is Chutusa, right? So assisted reproductive technology, you should be knowing that what happens in ZIFT, in GIFT. Okay, okay, where do we put the embryo if it is of 8 blastomeres? Okay, if it is of 8 blastomeres, you put it into fallopian tube. If it is more than 8 blastomeres, you put it into uterus. So if it is more, then you put it here or you put it in the fallopian tube. So you should be knowing about this thing. Okay, now IUDs, intrauterine devices, very much important. Out of all the contraceptive methods, this is considered to be very much important, and over here the most important one is hormonal IUD. I'm not saying others are not important, but this is very much important. Please go through it once. Okay, one extra time. When you go through others, go through it one extra time. Then vasectomy and tubectomy. Okay, you should be knowing what is cut in vasectomy. Vas difference is being cut. Okay, in tubectomy, what is being cut? You should be knowing it's fallopian tube which is being cut. Works? All this thing important, and you can study it. And one more thing, you will be asked, population will be increasing when maternal mortality rate increases or decreases. So mortality means death rate. Mortality means death rate. Okay, it means death rate. So if someone's mortality rate is increasing, obviously population will be going down because deaths are increasing. So you should be knowing about mortality rate. So if they are asking, population will increase if maternal mortality rate is growing high. So if it is going high, that means population will decrease rather than increasing. Same thing, infant mortality rate increases, that means population is increasing? No, it is the opposite. Infant mortality rate as in death rate is increasing, that means population will be going down. So you should be knowing about all this thing. Okay, so that is about reproductive health, and I hope please cover all the contraceptive devices for sure, but I am giving this as an extra thing because so many questions have come from this hormonal IUDs, okay, and asked about how do they work exactly. So you should be knowing about it. So that's all, my dear little children. I hope that I have covered all of it, and this is the entire syllabus for your whole biology 12th grade. Okay, in that you can tell me which are the chapters which are being added in your list, and if you want me to tell the important topics, I can tell it to you as well. Yeah, works? Cool. So that's all, my dear children. I hope that you are happy with the video, and I hope that you are not tensed, very much tense. Please be motivated. Please motivate yourself. Be in the positive sense that you can do whatever you are trying to do. Give your best. Okay, give your best, and there is time to change. If you think that you didn't do really well, you can do it the next time. Always there is a next time. Remember that. Do not be disheartened by the fact that, okay, this exam didn't go well, I'll mess up in the next time exam also. No, it's not like that. If this exam didn't go well, but I'll try for the next exam, I will do well in the next exam. That should be the motto, right? So that's all, my dear children. I wish you all the best and stay happy. Bye bye.